Thanks again for having me. So today I'm going to talk about initially our primary work, which is how we harness digital medicine and other related technologies for healthcare. But I'm going to conclude with some of our more recent work moving towards health and nutrition and food based on lessons learned from medicine. And so hopefully we'll have a fun time exploring some of the work that we are doing. I'm going to start off actually with a brief introduction to what N1 and Wisdom, or the Institute for Digital Medicine, are, and explore more in the healthcare side. What are some of the challenges with optimizing healthcare and therapeutics? Use a couple of case studies, but then move towards how we are harnessing these lessons learned from medicine towards food and health. So I'm going to start things off with the discussion about what our institute is and the people that make this work and innovation happen. So at the Institute for Digital Medicine in N1, we develop technologies and we validate them in clinical trials. But even though we develop technology, our ethos is that technology alone cannot transform healthcare. And so how do we bridge ideation with real adoption? First things first, when I talk about things like AI from our standpoint, we are not using big data to train algorithms to then make predictions, right? So the work I'm going to show you is not in silico. It is not purely computational. Everything we do is driven by prospective, actual data that we have to acquire in a strategic manner to drive an optimization process. And I'll show you examples about what we mean. But again, technology alone is not enough. We have this amazing interdisciplinary team ranging from, in fact, the first person we hired, who is not an academic, but hails from the insurance industry, to help us understand right off the bat, how do we get our technologies outside of our walls? What KPIs do we need to meet? What type of behavioral sciences studies do we need, which we have an amazing team of behavioral scientists that we hired? How about healthcare economics? What KPIs or metrics do we need to prove so that we can actually deploy what we do and impact healthcare systems? We have people from NUS Business, from comms and new media to help us with recruitment campaigns for our clinical trials, and many other disciplines. All right, so it really is truly interdisciplinary, but we move beyond interdisciplinary collaboration, right? We do something what I refer to as interdisciplinary empathy. When we think about empathy in healthcare, we largely think about doctor to patient, right? But I think it's important to consider the empathy that must exist between the disciplines, starting with the languages of the disciplines, but moving towards what those in the different disciplines and what all of these stakeholders go through on a daily basis so that we can truly understand how to deploy. Yesterday, I was speaking with somebody who asked us when we were discussing one of our platforms, which I'll talk about, how long will it take for your technology to be implemented in the healthcare system? And I said, well, you know, I can give you the media version of the answer. I can give you the real answer, right? And I said, in reality, okay, in media, you say 5 years, right? You go to it numbers 5 years. But I think what sets our institute apart is this understanding that the time that it takes for us to implement what we do, we have absolutely no real say in that. We have to go into this understanding that the patients have to want what we do. The doctors have to want and believe what we do. The healthcare system has to be able to justify why what we do exists in the healthcare system. There's many things we have no real control over, except as we move through the validation process, we will figure out if we can belong, right? So it's really not our say. And having that understanding is a major part of this concept of interdisciplinary empathy, right? That we have to be able to understand what everybody goes through. And I'll give you some examples of that and how that helps us do what we do. All right, so let's look at some examples of what we do. So about just over 2 years ago, I published a paper in Science titled AI in Cancer Therapy. And the process of bringing a new therapy all the way to patients is treacherous, and it's comprised of many facets, including drug discovery, drug development, and drug administration. One big issue right now with the field of AI, a lot of it is one word, right? So people say, hey, you do AI. That means you just discover new drugs. I'm like, we actually don't do any of this, okay? We actually focus mostly on this part. And people, even my own team members, use the words discovery and development interchangeably, but they're very different parts of the therapeutic pipeline. For me, discovery is trying to come up with new drug targets or new drug molecules, but what we do with these molecules, how we put them into combinations, how we match patients to trials, and ultimately how we dose the drugs will have a profound effect on the outcomes for the patient, because there's something that we learned in human, by the way. Even good drugs given at the wrong dose, given with other suboptimal drugs, will have no effect on the patient. But that doesn't mean that these drugs cannot work for the patient, right? So what happens is when you give otherwise good drugs, but at the wrong dose even, there's this misperception that the drugs cannot work for the patient. And just by changing the dose alone, you can make a patient go from non-response to response. And what does this tell us? It tells us we are missing opportunities to treat patients who can respond. It won't work for everybody, I'll tell you that right now. But as you'll see with some of our data, it can indeed work. So it's not enough just to come up with a drug that we think is good. We have to use them properly, right? And this is where things like AI and digital can help. How we acquire data versus how much data is needed. All right, again, no big data from us. In fact, the data we need to do what we do, to optimize treatment, to dose, that data doesn't exist. All right, because first things, we're all different from each other. That we know. Everybody knows this, right? And so we don't take large amounts of data, train an algorithm to treat the next guy that walks in the room based off of 10,000 people's worth of data or more. And the reason for that is while we're different from each other, we are also different from ourselves over time, right? We evolve. And so we need a way to use only our own data to manage only our own care, which means I don't have any data until the person walks in and we prospectively acquire and calibrate the patient. Even when we do drug combinations, if we take large pools of drugs, there is no pre-existing clinical trial data of many of these drugs interacting with each other. So we have to go and run real experiments to build the data that we need. And it's not as much data as we think. And so the governing approach that we use is this kind of second order equation, which we discovered with neural networks many years ago, where you can correlate the drugs and their respective doses with some sort of quantifiable outcome, tumor size, viral load, something you can quantify to tell you ultimate efficacy in a cell line, in a mouse, or in a human. You can correlate all of that with this kind of second order map, or this digital avatar, if you will. And in order to populate this map, you just need a few data points calibrated to get this done. All right, we'll give some examples of that in just a second. All right, let's look at some quick examples here. A few years ago, a patient approached us. This patient was in the US, stage 4 prostate cancer, given 2 drugs, responding to treatment, but super toxic. The patient was going to quit the trial, all right? And this was their last option. We were able to get some of the data. We actually needed some dose changes to occur with this patient, which they happened anyways because the drugs were too toxic. We're able to plot this kind of second order map, or this avatar. And this told us actually if we drop the dose of one of the 2 drugs, the efficacy would actually go up. Or imagine going to an oncologist saying, hey, you know what? Drop this guy's dose, his efficacy will go up. They're like, no way, right? But the patient stepped in and was like, look, I'm going to quit anyways. Where do I sign, right? Patient signs. A week later, their PSA level, which is their cancer marker, drops to the lowest level they had ever seen. One data point, right? We just get one data point of that PSA level, put it back into the map. The map doesn't really change. The doctors are like, no way. We're not going to do this twice, right? Patient signs, PSA goes down even more, okay? At the end of the study, the doctor's like, we don't do this in oncology. We give a high dose and a fixed dose. We see if they respond. That's it, right? In our case, we're like, but the patient's changing. The tumor's changing. We may have to dynamically dose to keep this patient optimized. Not long after that, patient in Singapore heard about this study, approached us. This patient at Tan Tock Seng, every time they were getting a dose of any drug, immediately put back into the ward, readmitted. Their liver enzymes are through the roof. They're in danger of liver failure and death quicker by liver failure than by cancer. After we got all the approvals, we started calibrating the patient with variable dosing. And even before we were able to start optimizing their dose, we found a response for the patient. Low dose, off-label drug, and you can see here, their tumors start cavitating, they start responding immediately, right? And we didn't even have to start optimizing. I said, look, is the patient happy? They're like, yeah, the patient's not rewarded, the quality of life is good, the doctor's happy. I'm like, let's just hold off. Let's keep them on this dose until we maybe have to escalate it, because this patient's at risk of saying, look, I'm done, right? If they're rewarded again, way beyond standard progression-free survival. Now, this is improving efficacy. About 8 months ago, we had a patient at NUH, stage 4 colon cancer. This patient's actually not responding at all, right? They're given a second-line combination therapy, no response. Cancer markers just keep going up. We calibrated a little bit, gave some variable dosing. Again, we dropped the dose at the end, and their cancer markers flipped from not responding to responding. Very different than here, which was responding, super toxic. Let's drop the dose to improve the response. This other patient is from no response to response. Don't change the drugs, just change the dose. Good drugs, wrong dose will not work. It's a misperception that the drugs cannot work. Again, our goal is not to low dose every cancer patient out there. Most patients still need the higher dosing, but our goal is to capture more responders when there's an otherwise misperception that they cannot respond. Okay, that's for the dosing. And I'll add that we have a team of healthcare economists. In addition to doing efficacy studies, we can actually start doing economic analysis. And we're seeing for some of these patients, we're saving them thousands of dollars a month, and also saving the healthcare system money and with better outcomes, right? So these are things we can start looking into now. All right, that's dosing, right? Using this quadratic avatar to do dosing in about September of 2019, okay? 2019, September, I say, hey, look, we can do optimization. We can also make new combos, not just dosing, but we can make cool combinations. And we were doing a lot of cancer, but I was like, let's go to infectious diseases, because like, this field needs way more attention than it gets, all right? So I convened a team of engineers. I worked with KPMG Singapore, with some global health security surveillance experts, and US business who do operations, developed a platform called Identify. Identify, again, is not big data. We run real experiments on live virus. And actually, one of our conclusions is that if there's ever a pandemic, we're gonna need quick ways and cheap ways to find better drug combos, all right? Because why? Healthcare systems will be strained, and industry will be either closed or also strained, supply chain issues, all of the above. And this was in 2019, okay? So we actually ran this experiment on at the time, a different RNA virus. There was no COVID, right? And then once the live virus was eventually isolated, we ran this on this, and then we quickly found that we could help clinicians prioritize which combos or drugs we should think about more, which ones we should think about less. Because different from oncology, we don't have time in this case, right? If you give a patient the wrong set of regimen, that's it. If it's not gonna work, things are already getting worse by the hour. We need to help doctors not only find which combos are good, but which combos they can maybe avoid, all right? So we did this in 2020 with the original wild type virus with a wonderful collaborator here. We repeated this in 2021 with the advice of doctors from NUH, NCID, and other hospitals. We picked a new set of drugs that were available in tablet form, right? We're going over 2021, looking for community deployable therapies. I won't go over this in detail. This is a workflow we developed. By the way, we were able to implement this workflow during circuit breaker, right? 2020, we design some experiments, we transfer the plates, live virus is dropped into the plates. We're effectively crowdsourcing live virus to tell us which combos are good, which ones are bad. We can predict some new combos, and then they give us the data, and then we revalidate it again with another experiment. 2 weeks, the whole thing is done, okay? And so we did this in 2021 with new therapies, all available in tablet form, came up with some new combos. At the time, we didn't have the Pfizer pill yet. We had the molnupiravir, the Merck pill. We found combos which showed they stayed true. They're still effective in the beta variant and the Delta variant, right? This is just efficacy of these tablets. And then we actually just finished 2 weeks ago an Omicron study. So we actually re-optimized for Omicron. And then it's, it's interesting. I'm reporting this today, and then today is when like everything lifts, right? But what's interesting is this is a 2 to 3 week study for the newer variants, right? It's something we're just going to keep running. Right? We're going to keep running this because it's fast, it's real data. The clinicians believe the data. And by the way, it's not only applicable towards COVID, right? We're doing this for antimicrobial resistance. We're doing this. We're going to start a study for dengue, I don't know if some of you saw, we've got more dengue cases now than all of last year combined. And dengue's always been a challenge to address, right? And so this is something that's easy for us around. We have a lot of data in the AMR space already, right? And so we're learning a lot, and we're just going to keep this going, right? It's part of our epidemic AMR pandemic preparedness platform that we're going to keep running. All right, final part. All right, so I've shown optimized dosing for drugs, optimized combo for drugs. Now let's talk about digital therapy, right? What if you don't have any pills you want to use? Let's talk about cognitive training. All right, this is a video game developed by NASA. It doesn't look very interesting, by the way. Okay, but it's a multitasking tool, right? Trying to hit the target here, manage some dials, respond to audio commands. What if you imagine this is a 3-pill intervention, but instead of modulating the dose, you modulate the difficulty? It's the same concept, right? And so we took 28 healthy subjects, ran them through this and scored them. And you can see over time, people get generally more accurate. Their reaction times generally get faster, but everybody's totally different from each other. Then we thought, okay, if we're going to modulate difficulty, does difficulty help people improve? And the answer is yes, right? High intensity, people get better faster compared to low intensity. So we know things change, but you also don't want to overtrain people either with their cognitive performance. So then we started dynamically changing the difficulty to parse out everybody's individualized performance over time. Some people thrive at high intensity one day, then they thrive at low intensity the next day, and it changes dynamically. And for many of these patients, these maps animate, right? These avatars change over time, so we can dynamically adjust their difficulty to optimize their outcomes. After that was done on healthy subjects, we received support from the Singapore Cancer Society to run this, what we call digital therapy. It's a worldwide field that's emerging, to run what we call the Cortex trial for brain cancer patients. Okay, and so we actually initiated that trial about 6 months ago. We're on our 6th patient now, and the game looks a little different. We brought in some animators from Lucasfilm, ILM, and co-created a new interface with the users, right? We got their feedback to drive better usage. We work with seniors, healthy subjects, brain cancer patients. We're expanding to the 30 to 40 crowd for some preventive studies as well. And now we're actually changing the game entirely. We're going to make new games as well to make them sustainable. But we're seeing patients perform their best through our AI recommended intensities. And it's all done on the back end. It's completely automated. We have a behavioral sciences team, Vivian and Smriti. What they do is they go and interview our users through a separate approved study to gauge feedback. We get feedback from doctors, nurses, everybody, to ensure that we can implement this properly. All right? Okay, so dosing of pills, combinations of pills, dosing of digital, final part of the talk. We've harnessed our approach with real experiments to optimize combinations. And we optimize combinations to produce quantifiable impact in living systems. The living system could be shrinking a tumor, preventing COVID or coronavirus infection of healthy cells. Can we apply this towards plants, right? Cell-based meats, things like that, because those are biological. And can we optimize how we address, these indications? And so everybody knows this better than me, right? We're moving in this field, food production. There's the global population is leading, and wars and viruses. Many things are contributing towards challenges in food security. Supply chain. When we did the COVID study in 2020, I'm sitting at the Shangri-La on a staycay with the family, talking to a former CDC epidemic intelligence officer. I went to college with him. And again, this is like February 2020, right? March, something like that. All I know is the next day I got kicked out of the hotel because it got turned into a quarantine facility, right? And so my buddy and I weren't talking about, we're designing the first experiment for Identify. And we actually were only talking about supply chain because I was like, give me 10 drugs you as an ID doctor would use for COVID patients that are repurposed. And he's like, Dean, I'm going to give you drugs, but we have to think about supply chain. Because if this thing gets way worse, you're going to have tons of drugs that could work, but you'll never see them. There's going to be trucks sitting at a port, but no driver, right? Or nobody driving the boat to get the drugs or flying the plane. So we spent, after we did that whole experiment in 2 weeks, we spent 2 months studying supply chain. Same deal with food, as we know. All right, so food yield. There's a lot of work in the field, right? Precision agriculture, genetics, and there's also titration-based modulation of soil engineering, what ingredients to put in. There's also anecdotal. Everybody has secret recipes from their families and what you can put into plants to help with yield. And it's the same thing with medicine, actually, right? Maybe with some more clinical trial-based evidence, but it's, hey, I've got the best ingredient, right? Eggshells, tapioca pearls that are dried out, rice, all kinds of stuff, right? And so we said, look, how about we run a systematic study to do some optimization of different ingredients, all right, to see how they interact, if there's any unexpected interactions. But can we take ingredients that are sustainable themselves to boost yield, but also not compromise nutritional content, right? You can grow a ton of plants, better yield, but if the nutrition content drops, there is no delta, right? And so we actually had fun with this. We ordered a bunch of these click and grow so we can have wells, much like we handle wells of cells. And we started with red spinach because we wanted to do a proof of concept where we can get quicker yield. And so through some of our initial studies, I can't share the ingredients yet. We're going to file some IP in like a month. You're going to find out. We're going to put everything out there open sourced. We selected a few ingredients with the help of some food scientists that they were interested in. And we were actually able to optimize combinations to increase yield as high as 30%. Okay. And we talked to many of the companies in Singapore about what they were hoping to achieve. And again, this is a first pass because maybe we could do even better if we try other ingredients, right? One thing is we can't optimize as quickly here as we do for like COVID studies, right? Because for that cell lines, we get quick turnaround. Red spinach, 6 to 8 weeks. If we want to do something like durian, it's going to take a long time, but we might try it, right? We're talking to rice growers. There's many things we can try, but workflow, setup, real experiments, real plants. And then, one thing to mention, again, not in silico, no big data. We prospectively acquire it and we experimentally validate everything, right? And so these axes are normally labeled with the different ingredients that we look at. And we can look at these interactions in terms of, hey, if you tailor the concentrations this much, can we drive yield up? And the answer is yes. And we're seeing this, right? And typically, and this is the same as with medicine, clinicians don't have this data, right? Where we show them like, hey, if you dose this way, you're getting no synergy at all, right? You actually have to dose this way within this area to actually drastically boost synergy up. This is another way to look at these maps. Ingredient 1, ingredient 2, and to add a multi-dimensional understanding of how combos interact with each other to optimize yield. But then the last question is, how about nutritional content? To sum it all up, we're finding that with less ingredients than we think, nowhere near their highest concentrations, we can have zero sacrifice on nutritional content. So using less to grow more with the same nutrition. And it's the same thing with medicine. We're actually seeing the exact same thing. Less drugs combined, not 4 drugs combined, but 2 drugs combined at doses lower than we think. The tumor actually shrinks less or shrinks more. It's a smaller tumor, right? And so that's what we're seeing, right? That's the real outcome that we want, using less to get more. So I think I'm just a little bit over time. Summary, drug optimization is not the same as drug development. I think this applies actually to food ingredient development and food ingredient optimization. We can titrate, right? When COVID happened, people were dropping any drug they could on a dish and they're like, oh, this drug is going to work. But the doses were like the equivalent of a brick in a person, right? And our work only looks at relevant dosing levels that can be tolerated by a human. And most of those drugs disappear at that point. Same thing with food, right? It's important. Food, we have a little bit more leeway, right? We can keep driving the amount up, but there comes a point where you keep upping the ingredient, it doesn't really help anymore, right? How much data versus how the data is acquired. By the way, big data is still important for many things, but for interventional, where you design interventions to go in to shrink a tumor, up the yield, you probably don't need as much data as you think, but you need the right data, all right? So lessons learned, right? I think that what we've done with medicine, we can apply to many other things, and we're doing all kinds of new food-related studies now. In terms of the broader scope, tech alone can't transform healthcare. A lot of this is driven by empathy, right? You see a lot of maps of digital avatars, but the final thing I'll show you is, or I'll discuss is, we recommended a low dose for a patient several months ago, and it goes through this pipeline of approval. And actually, the pharmacist contacted us and said, "Hey, look, I got it. Right? You want the low dose? I'll apportion the regimen. Just so you know, the way that this drug is aliquoted, if you give him this low dose, he actually needs to take more pills. Okay, because if you want 5 milligrams, it's 5 1 milligram pills. If you give him a 6 milligram, I can change things up a little bit, less pills. This patient never finishes their medication. I know this patient." So if you really want to go this route, you have to be prepared for the fact that they're not going to consume all 5 milligrams. They might take 3. They might take 0. Just a lot of pills in front of them. We don't think about that as engineers, right? But we need the pharmacist to tell us that. Costing of the medication, right? What people are going through their daily lives. In certain health systems, the nurse is the point of care. They're the ones that get clearance, but they're the ones that actually are the tip of the spear. We have to understand the whole workflow and to engage with all of them, right? And so when we say interdisciplinary empathy, what are the disciplines, right? And so it's certainly beyond engineering, beyond medicine. We're actually building an arcade for our digital health solutions, working with designers to do this, user interface, user experience, econ, ops analytics, regulatory policy, all of the above. The earlier we think about these challenges, I think the more streamlined bringing them into reality will be. So with that, I think that's the end of my talk. I want to make sure I acknowledge everybody who kindly supports this work and an amazing team of collaborators, including Jason in the room as well. We have projects together. So thank you so much.
Okay, thank you for your fabulous presentation. It's really interesting. A lot of thoughts. I think there was one question here.
Sure.
So was there a question? Sorry, I may have misunderstood. No. Well, maybe I can ask a question. So this is really fascinating. So how would this transfer to personalized nutrition? Because that's also NS1. And do you have any thoughts of that? Because there's so much talk about the individual diet for the individual. There are so many factors that go into this. You say this is not big data, this is not in silico, but do you have any hypothesis on that, if that could be used?
Sure, that's a great question. And I wanted to thank the organizers for putting the meeting together, and I apologize I can't be there in person. Yes, so we're doing a little bit of work in this space already. I think first things first, in personalized nutrition, I think we would need to work with, whether it's a clinical team or a nutrition team, to establish quantifiable endpoints that can be longitudinally assessed. And then what we would do is we would select the inputs, right? So we know the outputs, if it's going to be blood pressure, heart rate, sodium levels, et cetera, something that we can acquire dynamically. And then the inputs could be specific macros or other inputs that are deemed to be capable of being dynamically modulated. And what we would do is we would enroll a subject and we would give them variable dosing or concentrations of these inputs and measure their endpoints in time. And from that information, we would then craft one of these digital avatars. We can optimize multiple outputs at the same time, provided we can optimize multiple inputs. The more inputs we want to optimize and dynamically modulate, the more data points we will need. But the thing with N of 1, as you're absolutely right, is that because we're so dynamic from ourselves over time, that's why we would basically only use an individual's own calibration to modulate their own personalized nutrition. But again, if they're assuming they're a healthy subject, we may have more time to calibrate that user before optimizing. Very different than, say, a patient where we may have limited time to calibrate. But again, all the data is prospectively acquired. But it's something we are starting to work on, both for diet as well as physical performance and exercise as well.
Thank you. Thank you very much.
Thanks so much.
There's one other question. Can we have the microphone there? Thank you.
Yes, thank you very much. It was a really interesting talk. And I just was wondering how realistically we can apply this kind of technologies on the, population base. It seems very, very good to have it on individual basis, but I think that to have a real impact for the whole population, how realistic that would be in any different countries.
That's a great question. I think that one thing I'll mention is the resources required. So implementing this, first of all, does not require any supercomputing. In fact, a bulk of our optimization work can be done on a laptop. In terms of, so scale up wise, I mean, we can have one person handling tens plus, 100 plus or more subjects. I think the main thing though is still the logistics. So we just completed a pilot trial, a larger pilot trial for oncology. And we reported that at the ASCO meeting, American Society of Clinical Oncology. And I think right now it's more of a logistics thing. I think resources wise, it's feasible. The final thing I'll mention is that it needs to ensure that some degree of longitudinal monitoring is possible. So some indications will be easier to do than others. And right now we're trying to kind of find the optimal indications to go after so that it can help as many people as possible. But scale up is definitely possible, but easier for certain indications, doesn't require high powered computing. And so we're working on scale up right now. For some of our trials, we worked on hundreds of people, but again, it depends on the indication. Thank you so much.
All right, well, thank you very much. Dr. Ho, we need to move on. This is very interesting. So if there's any further questions, I think we have the contact details of you, Dr. Ho. Thank you.
Thanks again. Have a wonderful meeting.
Thank you.